Good afternoon, everyone. Welcome and thank you for joining us on this beautiful fall afternoon for this special lecture and celebration of the 2023 Rohlf Medal. My name is Sarita Bhatia, and I am the interim chair of the Department of Anthropology. And I'm so pleased to be able to host this event as part of the Provost Lecture Series. I'd like to first acknowledge the support of the Provost Office, the College of Arts and Sciences Dean's Office, and the Departments of Anthropology and Ecology and Evolution for their collaboration in this event. I'd also, of course, like to thank Jim and Janice Rohlf for their dedication to Stony Brook University and for making today's event possible. Our colleague Jim is a distinguished professor emeritus in the Department of Ecology and Evolution and a research professor in the Department of Anthropology. For those of you who don't know the history, which I am still learning myself, I'd like to give you some background on the Rohlf Medal. It was created in 2006 by Jim's family in honor of his 70th birthday. To date, 6 experts have been honored with the Rohlf Medal, 2 of whom are alumni of the PhD program in the Department of Ecology and Evolution. So thank you so much for joining us as we name our 7th expert who will be honored with us today. I hope you enjoy today's celebration and presentation. At this time, I'd like to introduce Jim Rohlf, who will share his own remarks about the Rohlf Medal. Jim received his PhD from the University of Kansas in 1962. He's a fellow of the American Academy of Arts and Sciences and the American Association for the Advancement of Science. Jim has lectured at and organized numerous international workshops on geometric morphometrics. So welcome, Jim.
Thank you. I thought I'd just give a little bit of background. In previous years, I've tried to minimize what I have to actually say. In fact, I think I said almost nothing in previous years. As Sarita mentioned, this was established 2006, but I should say the idea was established then. I should also honestly say that it wasn't my idea. My wife, Janice, it was her idea. I thought, well, this sounds pretentious, but might be a good idea. But I agreed to it finally because I thought it would be useful to help benefit the field of geometric morphometrics. It would bring attraction and attention to the field, hopefully bring in new people with new ideas. And it is a field that is still evolving quite a bit. It evolves as the technology changes. Originally for morphometrics, the study of shape and shape variation, if one could only measure things with a ruler, then you would describe shape by saying maybe length-width ratios. As we could have devices that would digitize coordinates of things, take digital photographs. Now we have MRI, CT scans, laser surface scans. You now can capture really the whole image or the whole volume of an organism. That requires new and much more sophisticated statistical methods. And that's what's ongoing and hoping to track in the field, people who will have that mathematical knowledge to be able to do proper statistical analysis of this complicated types of data. So I'm looking forward to the fact that in the future we'll attract some new smart people in with new ideas. The other thing I mention is that the scope of this, I think Dean will partly say also, is that the winner is selected by a scientific selection committee made up of people with wide experience in morphometrics. It's purposely intended to be a committee with people with rather broad knowledge, both mathematics and biology, to be able to deal with winners who will be using advanced statistical and mathematical methods, as well as applying it to interesting, important problems in biology. Dean will announce mention who's on the committee. But I'll just say that I think the committee did a very good job this year, and I'm looking forward to his lecture. Thank you.
I'll bring this back down. I'm not quite as tall as Jim is. Good afternoon, everyone. My name is Dean Adams. I'm an alumnus of Stony Brook. I received my PhD in 1999 in ecology and evolution, and I had the honor of serving as chair of the Rohlf Medal Committee this year. Our committee was comprised of myself, let's see if I get them in alphabetical order, Fred Bookstein, Eric Delson, Benedict Hallgrimsson, and Joan Roughgarden. And so we had a group of individuals that span a wide breadth of expertise, and we went through all of the candidates, and we came to, I think, what is a fantastic choice. And I will just summarize briefly what we decided. So the general consensus and the strong consensus was that Professor Mitteroecker's work covers a broad range of biostatistical and morphometric approaches that attack biological questions from multiple points of view. And I'll expand on this in a few moments. His work spans advances in morphometric representation, the development of new theoretical and analytical approaches that further empirical research, and his own broad application of these tools to address and advance questions in biology and in anthropology. And so it was for these reasons, for the breadth and depth of his contributions, that the Rohlf Committee selected Professor Philipp Mitteroecker as the 2023 Rohlf Medal recipient. Congratulations. At this time, I'd like you to come forward, and Jim, if you would please.
Thanks a lot.
Looks good. Congratulations.
Thanks.
So, before I turn the reins over, let me expand on that a little bit more and give you all a bit of context. So, we're here to celebrate Professor Philipp Mitteroecker's receiving of the 2023 Rohlf Medal for his excellence in morphometric methods and their application. Philipp received both undergraduate and doctorate degrees from the University of Vienna, the latter in 2007. He then worked as a postdoctoral researcher at the Konrad Lorenz Institute for Cognition and Evolution, and then joined the faculty at the University of Vienna, first in the theoretical biology department, and now he's a full professor in the Department of Evolutionary Biology. As I alluded to, his work spans an incredibly broad range of both quantitative and biostatistical questions that are germane to morphometrics, quantitative biology, and the empirical disciplines that they affect, including evolutionary biology and anthropology. He describes himself as, quote, an evolutionary biologist, anthropologist, and biostatistician. And not surprisingly, his major research contributions are at the interface of these arenas. Through more than 100 publications, Professor Mitteroecker has made impactful contributions that have altered our understanding of biological anthropology, of developmental biology, and of evolution. In my opinion, his work has pushed the boundaries of our biological understanding of morphological variation, as well as providing mathematical and theoretical underpinnings of how we may quantify and evaluate those patterns. So on the more conceptual side of his work, Professor Mitteroecker has contributed numerous methodological and analytical advances that currently lie at the core of our 21st century approaches to morphometric analysis. A partial list of these contributions includes the development of three-dimensional semi-landmarks for quantifying 3D curves and surfaces, the proposal of size-shape spaces for elucidating growth trajectories, methods for understanding patterns of morphological integration, and approaches for contending with pesky things like missing landmarks, because of course our data never come in nice, neat packages. He's also delved deeply into measurement theory and investigated how the topology of our phenotypic spaces themselves can actually impart constraints on what we can and cannot say as we're trying to elucidate trends in our data. And in this regard, his work on affine invariances was in fact instrumental to understanding how to interpret differences of trajectories and changes across phenotypes, depending on the sorts of phenotypic data that were under scrutiny. And this really shed light on that aspect of our work to make us think more deeply about how we marry our data with our methods. In addition to theoretical contributions, Professor Mitteroecker, his work in evolutionary anthropology is equally impressive, and his many contributions to this arena have advanced our understanding of the evolution of both human and non-human primates. I'll give one example, which is actually rather early on in his career, but it's one that's impactful, and frankly, I teach in my classes. He examined evolutionary growth trajectories in humans and related primates using unique procedures that demonstrated how similarities may result from so-called common allometric components that then subsequently later in development diverge to result in the unique morphologies that we see across taxa. In fact, his contributions to evolutionary anthropology are as numerous as are his theoretical contributions and have had a large impact on how we understand the origins of our own group. Recently, Professor Mitteroecker has turned his attention to the so-called obstetric conundrum in an attempt to shed light on how human health practices regarding childbirth interrelate with evolutionary selective pressures on the human pelvis. Indeed, this aspect of work he touches upon will be a delicate subject of the measure and mismeasure of man, which I note is part of our title today, so I hope to hear more about that. And finally, I want to give just a little personal anecdote. I've known of Philipp's work for many years, and we've communicated digitally for many years, but recently we finally struck up a formal collaboration, the first of which is now submitted, and I can say without hesitation that he is absolutely as intense, as creative, as thought-provoking as his papers imply. And it has been a pleasure to work with him because looking at things from his perspective has helped enhance my own. And I hope it's been reciprocal, and I can't wait for that collaboration to continue for that very reason. So, in light of these many contributions, I am happy once again to welcome Professor Mitteroecker to the stage so we can hear about his work. Philipp. Congratulations again.
Thanks so much, Dean, for these very nice words, and also to Jim and to the committee for this award. It's really a great pleasure and honor. Today I'd like to speak about the mismeasure of human evolution, but let me first say a few words how I became a morphometrician. It was in 1999 when you finished your PhD that I was an undergraduate or just graduate student, and my colleague and friend and longtime collaborator, Philipp made me aware of an advertisement in the anthropology department in Vienna announcing a guest professor named Fred Buxton giving a course on morphometrics. And he said, "Look, Philipp, this sounds like really cheap and easy credits. Let's do that." And I said, "Wow, morphometrics, that sounds so terribly boring. I don't want to do that." So, but finally, I was convinced by the promise of some cheap and easy credits. So we did it. And well, both of our expectations turned out to be wrong. It was neither easy nor was it uninteresting. So I got stuck with morphometrics ever since. And many of the developments that you have just mentioned, I did together with my colleague Philipp Gunz. And Fred was finally both our master's advisor and also PhD advisor, and we did many research projects and papers together. In this very first course of Fred, we also met Katrin Schäfer, some of you might know, and she was back then a fresh assistant professor, and at the very first lesson I asked her, "Do you have any idea what Fred is talking about?" And she said, "No." And so I understood basically nothing what he said, but it seemed interesting, and it motivated me to learn the things that I didn't understand, to go into the math and statistics that I had no idea, obviously, back then. And it was the start of an interesting research trajectory, and I'm very grateful to all those. So here's a picture of Philipp Gunz, me, and Fred together. Back then I used to have a different hairstyle, but I inherited some of Fred's influence with regard to that. Fred was also the first recipient of the Rohlf medal. A year later, in 2000, Dennis Slice was a guest professor in Vienna, and he held a course there. And since then, he regularly visited Vienna to teach there, and he passed away way too early, and we miss him a lot there. In 2004, I had the great opportunity that Jim came to Vienna and gave a guest lecture there on multivariate statistics. So it was really in the great opportunity and unique opportunity to have Fred, Dennis, and Jim as teachers, and that was very influential to me and really important. Other important Rohlf medal recipients in my life were Paul O'Higgins, who reviewed our very first paper together. And that was in Journal of Human Evolution, and he was one of the reviewers, and the other reviewers all said, including the editor, that this is really a crappy paper. So the biology is really bad. We have obviously no idea what we are talking about, but Paul insisted that the methods are important. And eventually we made it, and it became a well-cited paper, but in hindsight, the biology was really crappy, and I hope we learned how to do it better. Also, Benedict Helgrimsson was over the years an important supporter, and I'm thankful to all of those. And all three got, all they got the Rohlf medal except for Jim, but Jim is the Rohlf, so he doesn't need the Rohlf medal. Yeah, and meanwhile, in the years after that, I worked on a lot of different methodological and theoretical topics, and a lot of applied research in evolution and development. And in most of those topics, there was one way or the other, there was morphometrics or multivariate statistics involved. And I learned it the hard way that talks with a lot of formulas don't provoke a lot of enthusiasm. So I decided today that I will talk about an applied topic, namely the evolution of the human pelvis and childbirth. And it all started with my colleague, Barbara Fisher, who came to me almost 10 years ago and said, I want to measure the pelvis and study the pelvis. And we started basically with a simple question like allometric scaling. So how do pelvis differ and vary in relation to body size and things like that. And she got, and to basically to say something about the background on that, why that's interesting, because in the first moment I thought, okay, the pelvis, why not? But I was quickly convinced that this is a really interesting topic. Why so? Well, human childbirth, or say live birth in general, viviparity, actually is quite old, and it didn't only involve in mammals. It also involved in different lineages of sharks, amphibians, lizards, mammals, and so on, and it's pretty old. So viviparity dates back to the Jurassic, and the mammalian placenta and eutherian placenta dates back to the early Cretaceous. So there has been plenty of time for childbirth to evolve. Nonetheless, human childbirth is challenging, right? We know that obstructed labor, which means that despite normal uterus contraction, the baby doesn't exit the pelvis because it is somehow blocked, that this occurs in roughly 3 to 6% of the births worldwide. And this is a bit surprising, right? That something as essential to a species such as reproduction, childbirth, is so problematic in humans, right? Such a long and painful and often dangerous process. And it's really kind of an evolutionary puzzle how that came to be, how that evolved. And this is what fascinated me. And in my research, we especially focused on anatomical problems, anatomical disproportions. So cephalopelvic disproportion, meaning that the head is too large, of the head of the fetus is too large to pass through the birth canal, or sometimes also the shoulders that don't fit through. So altogether, I refer to that as fetopelvic disproportion. The fetus is too large to pass through the maternal birth canal. And cephalopelvic disproportion, the rate of that is actually surprisingly difficult to estimate, but reports range from 1 to 8%. A large US statistics reports that for infants weighing 3 to 4 kilograms, there is about a 2.3% rate of cephalopelvic disproportion. For heavier infants, that can go over 5%. So surprisingly common. From an evolutionary perspective, this implies strong selection, because without modern obstetrics, without cesarean sections and the like, there's a very high probability of maternal morbidity or a mortality associated with obstructed labor. So again, the question is why and how did that evolve? The immediate reason, obviously, is that in humans, the fetus, the head of the fetus is very large compared to the birth canal and also compared to other primates. So in other apes, usually the baby is much smaller compared to the birth canal. Not in all apes, especially not in all primates and not in all mammals, I will come to that, but at least among the great apes, humans are unique here. And it's clear that a large brain is essential to our species evolution, but the question is, why didn't the pelvis evolve wider, right? Why that? And a classic explanation, which is often referred to as the obstetrical dilemma hypothesis, is that if the human pelvis would be wider, involving a larger birth canal, that would be problematic for the biomechanics of upright walking. Yeah? That has been a very prominent explanation that has only been tested in recent years with very unclear results. So several studies said that there is no clear evidence for that, and I would say that remains an issue to be settled. But apparently, whether or not there is an influence on pelvic width on the energetics of locomotion, it's probably a small effect. So it might not be the main reason for that. Another reason why the birth canal didn't evolve to be wider could be pelvic floor stability. So you know that there is a muscular pelvic floor, and in upright humans, that pelvic floor has to carry weight, as opposed to quadruped primates, where the weight of the internal organs basically is on the spine and on the belly, not on the pelvic floor. So the idea goes back already to the 1980s, and there is quite some epidemiological work showing that pelvic canal width correlates with pelvic floor disorders, such as pelvic organ prolapse or incontinence. So women with a larger birth canal, where the pelvic floor has to span a larger area, are more likely to experience pelvic floor disorders, such as prolapse and incontinence, than women with a smaller birth canal. In a biomechanical model where we used finite element analysis to model the pelvic floor for differently sized pelvic canals, we confirmed that. So that a larger pelvic floor basically descends more and is more likely to experience some sort of failures. In a second biomechanical study, we also showed that not only the size but also the form of the pelvic canal is essential, because a roundish, a more circular birth canal, which gynecologists refer to as a gynecoid pelvis, which is said to be optimal for childbirth. So we will come back to that. So a roundish gynecoid pelvis, the gynecologists consider as best for childbirth, whereas the pelvic outlet, so the lower part of the pelvis, where the pelvic floor is basically attached, usually has an anterior-posterior elongated, so an oval form. And we showed that this oval form is supportive for pelvic floor stability, whereas a roundish pelvic canal shape is basically weakens the pelvic floor stability. Pelvic floor disorders are relatively common in modern populations, but in Western populations, that's a severe inconvenience. But without proper hygiene, without antibiotics, the risk of serious infection is relatively high. And pelvic floor disorders, even though now it is mainly affecting women of higher age, also occur in women during the reproductive period. And data clearly shows that this reduces, especially without medical treatment, survival rates and the number of offspring. And actually in sub-Saharan Africa, it is still a very serious problem that women with chronic incontinence face strong social ostracization. So what we see is that the human pelvis probably evolved in response to numerous and partly antagonistic selective forces. Obviously, a large birth canal is good for childbirth, but it's detrimental for a number of other functions. And so coming back to the morphometrics, so my study on, or my research on all these topics started really with a morphometric question. Barbara came to me and said, how can we study, how can we measure and study pelvic form variation? And what she had is she got data with linear distance measurements on the pelvis, of the pelvis, and we studied it very intensively and didn't find anything. That was a bit of a surprise because we really tried hard to do some classic morphometrics and really didn't find anything. So it was clear we need some landmark data, we need geometric morphometric data. And actually Barbara found that, we know actually also from studies of simpler structures like crania that using distance measurements can be less effective than working with landmarks, even though I've never experienced that difference so strong there. And I think one of the reasons is that really the overall geometry. A mathematician once said to me that to him, the cranium is just a sphere. Topologically, right? And if we ignore little holes, that's kind of true topologically. The cranium is basically a sphere, like a soccer ball, right? But the pelvis is a torus, it's a bagel or a donut, if we ignore the holes. But of course, there are larger holes here. So we have really a more complicated topology and many details that matter. Yeah? And I think that really makes accounts for the reason that with distance measurements we were so unsuccessful. And so Barbara managed to get landmark data. And actually, that's interesting in itself. So she got data from Herbert Reynolds, who with colleagues measured pelvis. They collected hundreds of 3D landmarks on pelvis in the 1980s, so relatively early, back then for the Federal Aviation Administration to improve crash test devices. So no real evolutionary or anatomical question. And so she contacted them if we can use the data, and they were very happy to share it and people do something like that. So it took actually quite a while to clean the data for modern biological questions. And the data we had were from skeletons collected basically in the early 20th century for the Heyman Todd collection. And there were really many anthropometric measurements before skeletonization, and after skeletonization, they took the 3D landmarks of the pelvis. We ended up with a sample of 126 3D landmarks for 99 million female pelvis. And many of the analyses I will show you now are based on these data. And the first study that we did was basically a study of allometric relationships in the pelvis. And that sounds a bit maybe boring and traditional, but I think the questions behind I liked. And they sound maybe a bit strange in the first moment. So the question is, or the question that we asked is, what about the risk of obstetric problems, of obstructed labor? Is that the same among all women or not? And well, from the gynecological literature, we know that there are a number of certain risk factors, including maternal age and constitution, all kinds of diseases such as obesity and diabetes, but also and foremost, maternal and paternal stature and fetal head size, right? So after all, what really matters is that the fetus, especially the fetal head, fits through the birth canal. And we know from the literature that shorter women tend to have higher risk of obstructed labor than taller women, because short women might also have a large baby, especially if the short woman has a baby with a tall father, so that these women face a higher risk of obstructed labor than taller women. And also, if the baby has a large head, the risk of obstructed labor is large. And we don't have, for this data, we don't have the head of the baby, but we have the head of the mother. And head dimensions, including intracranial volume, has a pretty high heritability. And so we took actually the mother's head dimension as a proxy for the baby's head dimension, because on average, they are pretty related. Yeah? There are studies on that. And so we expect stronger obstetric selection in short women as compared to tall women, and we expect strong obstetric selection for women with a large head as opposed to women with a small head. Yeah? And so we expected that the optimal evolutionary compromise between these opposed selective forces that I mentioned below might actually not be the same for all women, but it might differ between short and tall women. And so what we studied here is we basically took these landmarks, superimposed them, so we calculated shape coordinates and regressed them on body height in women. And what I have visualized here is an average pelvis for tall women and an average pelvis for short women. And you see there are some clear differences. You see that in short women, remember those who likely are under strong obstetric selection, the birth canal is more roundish, more gynecoid. So it's the birth canal shape that is expected to be better for childbirth than in tall women. And also you see that the sacrum here, the bone, is projecting outward in short women with the higher risk of obstructed labor. So it enlarges here the birth canal as opposed to tall women. So it's actually what we expected in a way, yeah? That short women who have a higher risk, in theory, of obstructed labor has a more birth-friendly geometry of the pelvis as opposed to tall women. We slightly see the same pattern in males too, indicating that there's probably a pleiotropic genetic effect here. When we regressed these shape coordinates on head size, taking body size as a covariate, so basically we studied the effect of head size on pelvic geometry independent of body size, we also see an interesting picture. We see that women with a large head here on the right side have the more roundish birth canal, so the birth canal better fitted for childbirth as opposed to women with a small head. And also we see that the sacrum is clearly projecting outward, enlarging the birth canal in women with a large head. So actually that fitted our expectations very well. We partly see this again in males. So we concluded that the integration of pelvic canal shape with body dimension might have been evolved to alleviate the obstetrical dilemma. And it presumably evolved in response to the correlational selection regime imposed by these manufacturers on childbirth. A second morphometric study, a pretty recent one, focused on the sex differences in the pelvis. That's also a classic field of study because the pelvis is the element of the skeleton that shows the greatest sex differences. So the female pelvis is larger than the male pelvis, not only in relative terms, but also in absolute terms. And that's the only part of the skeleton for which that's true. I carried out this study with my colleagues Barbara Fischer, Nicole Grunzer, and Eva Zaffarini, a previous master student. And we had here data both on humans and chimpanzees. So what we did is we compared the sexual dimorphism in humans and chimps. And this has been done before, mostly with classic morphometric approaches, basically distance by distance. But as you will see, that is not very powerful. With our approaches, we could find some quite interesting results. So here is a visualization of the average sexual dimorphism in the human pelvis, a very well-known pattern. We see that in males, they have a narrower, more elongated birth canal, as opposed to women. In women, the subpubic angle is larger, which enlarges the birth canal. You also see that the sacrum is shorter and more projecting outwards in women than in men. So that's a classic finding. When we studied the same dimorphism in chimpanzees, it looks kind of similar. You see that also here, the female pelvis in chimps has a larger, wider birth canal, a shorter pelvis, and a larger subpubic angle. Nonetheless, it's a bit difficult to compare that because the species differences are so strong here. So what we did is we took the sex differences from chimpanzees, so we basically subtracted male from female, took this vector of sex differences and put it on a human pelvis. So we deformed the human pelvis along the sex differences of chimpanzees, basically to undo the species differences, but compare only the sex differences. And if you look at that, the visualizations are basically identical. So they were scaled to the same magnitude here. I'll come to the magnitude in a second, but what we found is that the sex differences, the pattern of sex differences, and that's important, not if you compare, dimension by dimension, but the multivariate pattern of sex differences is virtually identical in humans and chimps. And that is a bit of a surprise because chimps have a much smaller newborn than humans. So one wouldn't expect that strong obstetric selection in chimps as compared to humans. And it is often, the large sex differences in humans is often interpreted as a human-specific adaptation. So why do chimps have the very same pattern? And here is a principal component analysis. On the left side, you see the chimpanzees and the chimpanzees and the humans. And so what you can see here is basically, again, in a multivariate sense, that the same pattern of sex differences in human chimpanzees, but a different magnitude. So it turned out that humans have about twice the magnitude of sex differences than chimps, even though the very same pattern. Yeah. So what we find is a conserved pattern of pelvic sex differences, but a different magnitude. And we think this is important. In fact, humans are not the only species with large fetuses. In primates, for example, there are macaques, there are gibbons that also have very large fetuses, but in mammals, there are many species with large fetuses. Some even have fetuses way larger than humans compared to body weight. And some species, such as guinea pigs, bats, have fetuses that are larger than their birth canal. So what we find here is that the pubic symphysis opens widely, but that's another topic. That's not possible in humans because if our pelvis would open like that, humans couldn't walk anymore. So in humans, that flexibility of the pelvic girdle is highly reduced to most other mammals. And so probably the problem of having giving birth to large fetuses is very old, in fact. Probably it dates back to early mammals or even amniotes. Yeah. What developmentally, the pattern of sexual dimorphism in the pelvis is determined by the spatial distribution of receptors, of estrogen receptors and relaxin receptors. And we suspect that this is, like most physiological or hormonal aspects, are highly conserved in mammals. And this is a really old pattern. But the magnitude of that, which is basically the trigger of that conserved mechanism, basically the overall magnitude of hormone expression is that this is variables. So we expect to have a conserved genetic developmental machinery of these sex differences, but a quite evolvable knob, so to speak, developmental knob that can turn off or on these developmental pathways. An idea that in evolutionary developmental biology is referred to as facilitated variation, that you have some conserved developmental pathways that have a very evolvable switch or knob to turn on or off evolutionary. And we think that this has also been important to human evolution when the human brain evolved and enlarged so quickly that the pelvis could keep track of that by basically simply turning that evolutionary knob. Another interesting morphometric question that has been asked by Helen Kirky and others is, given that under these evolutionary scenarios, well, females are faced by strong opposed antagonistic selection, which should lead to stabilizing selection, right? So as compared to men. So we would expect that, or Helen expected that women should actually show less morphological variation in the pelvis than men. However, she studied that extensively with classic morphometrics, distance measurements, and studied variances in distance by distance, and she didn't find any evidence for that. But I found this idea very interesting, and so I started studying that. This is still unpublished, but I went into that some time ago. But I started with a different expectation, because unlike most other bony dimensions, the pelvis undergoes a greater magnitude of shape change in females as compared to males, because in puberty, the female pelvis remodels more than the male one. So, and given that, we would actually expect that the female pelvis should vary more because it develops more than the male pelvis. So overall, we would expect the female pelvis to vary more than the male one. However, given this birth-related selection, it might be that some aspects of pelvic shape, not all, but those that are relevant for childbirth, might face stronger stabilizing selection. It's only some aspects of pelvic shape that show reduced variance in females as compared to others. And the question is, how can we study such complex variational properties, right? For this, I used the data introduced already before from the 19th century, as well as a second data set on pelvis from the 20th century, from the late 20th century. These data were collected by a former PhD student, Lukas Waltenberger, as well as some chimpanzee data collected by Eva Zaffarini. And we analyzed that by a method that I published with Fred some years ago, which we called relative principal component analysis or relative eigenanalysis. And the idea is here that you have multivariate data and actually two groups, two samples. For example, males and females, or two species, or between and within group variation. And you would like, in principal component analysis, most of you know that, we have high-dimensional data, and we look for linear combinations for directions in data space with maximum variance. Here, we have two groups, and we look for linear combinations or direction data space with maximal variance ratios. So we look for trait combinations where one group shows maximally more variance than the other group. And this is what we used here. And so if I would just pool all variables and calculate total variance of all variables for males and females, for the 19th century data, we found that, as I expected, females do show more variance overall than males. But in a second step, I tried to decompose that, right? Based on several ordinary principal components, I did this relative principal component analysis, relative eigenanalysis. And so you see here the first component. So that's a shape feature that shows about 4 times as much variance in females as compared to males. The second one about a bit less than 3 times, and so on. And you see the last one is a shape component that is less variant, less variable in females as compared to males. And this is actually the interesting, the candidate where we would find stabilizing selection in females as opposed to males. And this is a visualization of that. So this is the shape feature, the only clear shape feature that where females are less variable than males. And you see it's interesting because it's exactly the birth canal dimensions. It's basically the width of the birth canal, which is most decisive for childbirth. If you look at the other components here, these are just different shape features. So there's a lot of going on here, but the feature that is canalized in females is basically the birth canal dimensions. And so this is what we would expect here. When I project the chimpanzee data into these data spaces, so you see that this is the scribbled from before, and these are the variance ratios for these shape dimensions for chimpanzees. We don't find that pattern. That's also kind of reassuring because we know that in chimpanzees there is less obstetric selection, and so we might not find it, and we didn't find it here. Yeah. I also tried this for the 20th century data, and you also see here that different dimensions show different variant ratios. What I plotted here is the last relative PC. So it's again that shape feature for which the women showed less variance than the males, and it's different. And I'm not sure yet whether that's a finding or not. It could be because in this 20th century data, well, all these women had the opportunity of C-section. So obviously in the late 20th century, the obstetric selection, luckily, has been strongly reduced. So probably it might be that what we see here is already a change in the selective regime over these 100 or 150 years. Yeah. But that probably, that needs some more work, I guess. So, okay, so much about morphometrics on that. So we did some morphometric work on that, but there's not enough time to show you that. Rather, I would like to show you another basically theoretical work that we did, because there's pretty much an agreement, even though not everybody agrees, that it will be my last slide, but most people agree that the human pelvis has been subject to this antagonistic selection, right? The pelvic canal didn't enlarge more evolutionarily because that would have been, detrimental for other things. What I was wondering, how can it be possible that this relatively high maternal and fetal mortality basically is outweighed by some probably small advantages for a small birth canal, right? How is that possible? And so we actually developed a model which we called the cliff edge model for reasons that will become obvious. So I did this together with Simon Huttecker, Barbara Fischer, and Michaela Pavlitsev. And the idea is the following. So it's of course a gross simplification, I'm aware of that. So we represent these fetal-pelvic proportions by a variable called D for difference. And it's a difference between the size of the neonate and the size of the maternal birth canal. So D larger than 0 would indicate fetal-pelvic disproportion because the fetus is larger than the birth canal and doesn't fit through. What do we know about selection on this variable? Actually quite a lot. We know from the medical literature that a larger baby, a larger newborn has higher survival rates and is less susceptible to a number of diseases. So there's obviously selection for large neonatal or perinatal dimensions. We also know from the studies I showed you that there's selection towards a smaller birth canal due to the pelvic floor stability, maybe also due to the energetics of locomotion, but I would say that that's an open question. So after all, there is clear selection for a large D value, large babies, small birth canal. If we would plot that, so if I would plot that variable D here, so this difference between fetus and birth canal, and on the y-axis the individual fitness, we would see that fitness increases with D, with a large baby, with a small birth canal, until to the point where the baby doesn't get through anymore. Then, without cesarean section, there's a severe loss of fitness. You see that fitness function doesn't look normal as in many evolutionary models. It's a cliff edge, basically. Yeah? And the question is, what can do evolution about that? Most of these dimensions, and there's a lot of data on fetal and pelvic dimensions, they are pretty normally distributed. So the phenotype looks like that, but the fitness function looks like that. And what do we expect here? The fitness increases with a large D, so we would expect that the phenotype distribution evolves to the right. Babies become larger evolutionarily and the birth canal smaller, until some part of the distribution, some individuals fall off the fitness cliff, right? So the evolutionary stable state, so the evolutionary optimum, in a way, what is achieved is an equilibrium between the advantage of the individuals with large newborns and a small pelvic canal versus the few cases faced with obstructed labor. So the evolutionary stable state will be something like that, where some cases always, some percentage of the population always experience fetal-pelvic disproportion. Evolution can't do any better than that. Yeah? And it's also intuitively clear that the stronger the selection, so the steeper that line, the more individuals will fall off the fitness cliff, right? And given that we have some data on the rates of fetal-pelvic disproportion, we can actually do the opposite and estimate the strength of selection that yields that percentage of cases. And this is what we did. So we developed the mathematics of that and found that, as I said, these rates of disproportion are relatively difficult to estimate. But if you say they are somewhere in the range from 2 to 6%, maybe even a bit less or more, this would translate into what we call a standardized selection gradient of 0.06 to 0.16. This is, so there are, it's a bit unclear whether this is a lot or not, but there are many studies on the strength of selection for all kinds of traits and all kinds of animal and plant species, and they show that this is rather weak selection. Yeah? So what this model shows is actually that, at least my intuition was wrong, right? It doesn't need much selection for a small birth canal or for a large baby to yield that relatively high rate of fetal-pelvic disproportion. it's the fault of this asymmetric fitness function. Yeah? And it doesn't need much of selection to push that many individuals over the fitness cliff, so to speak. And well, again, this is just a very gross model, and one aspect in which we can make a bit more detail is that before that basically applied to female fitness, but in the population, of course, we have roughly half males, females. And you know, if in the most extreme case, we could argue that if the baby is too large, in the case of fetal-periodic disproportion, the mother might die, but the father could still reproduce with others. I mean, that's not very romantic, but in principle, that's possible. And in that extreme case, we would have a situation like this, where if D is that large, female fitness drops, but male fitness might somewhat continue to increase or to stay stable. And in that case, the situation is even worse, right? In other words, we would have selection for a large offspring or a small birth canal, basically in both sexes, but the obstetric selection towards a large birth canal is only there in females. So in that sense, the males also push the distribution here towards the right. And so in this situation, the observed incidences of fetal-periodic disproportion would require even a much smaller selection towards a large D value. So, and of course, the true situation is somewhere in between these models, depending on all kinds of social factors. And so, but the message is that it really needs not much selection for a large baby or small birth canal to lead to that cases of fetal-periodic disproportion. That's one insight. The other insight is what happens now. Today, luckily, very few women die, at least in the global north, during childbirth, because if the baby really doesn't fit through or there are some severe problems, they get a C-section, a cesarean section. Yeah? What does this to our cliff edge model? Well, the cliff disappears, right? Now, luckily, we have a situation like this. If the baby is too large, they still survive, luckily. So what do we expect? I mean, this was an evolutionary equilibrium, an evolutionary stable state, but that stable state has been disrupted. We would expect that evolution starts again. And that's a bit of a paradox, right? We face now actually less natural selection, luckily through modern medicine, but one would maybe at first sight think that this reduced natural selection would reduce evolutionary change, but the opposite is true. Because an evolutionary equilibrium has been dissolved and evolution starts anew in a way. Yeah? And the question is, I mean, is that fast enough to be relevant? And we estimated that because I've shown that we were just able to roughly estimate the strength of selection, the steepness of that curve. And cesarean sections, I mean, we know that for a long time, but really at a regular and safe basis, they are practiced roughly since the 1950s or 60s, so more or less 2 or 3 generations. So if we apply that model to these data, we would see that over these 2 generations, this model predicts an increase of the original incidence of fetal-periodic disproportion by roughly in the order of 10 to 20%. Which makes about half a percentage point. This is not much, given that we have C-section rates of 30 to 50% in the Western world. But nonetheless, that we find an evolutionary change as visible as that within 50 years in a modern society triggered by medical intervention is nonetheless surprising and astonishing. So what we see by that is that evolution is taking place in modern societies, which is, at least to many in the public, surprising, because most think evolution is something that happens over thousands or millions of years, but that's not true. The question is whether our results are true, whether the model is true, given the many simplifications of the model. And it's not easy to test this with epidemiological data. We tested that indirectly because we were able with that model to pretty exactly predict the inheritance, the intergenerational inheritance of the predisposition to dystocia, so fetal-periodic disproportion basically in cesarean delivery. So indirectly we were able to validate to some degree. So we believe that even though the exact numbers are not trustworthy, but the overall magnitude effect we believe is reliable. So finally, let me say a few words about the non-biological aspects of that. What I was referring to so far is basically the genetically determined parts of fetal dimensions, of pelvic dimensions. But of course, not everything is genetically determined. And also, we know that childbirth is also a social phenomenon with a lot of cultural and medical influences. And so clearly, after all, it is the match between fetal and maternal body dimensions that is decisive here. But these body dimensions are influenced by a number of environmental factors. So we do have a plastic component here, and there's also a lot of interesting research on that, and we also published on that, but I can't go really into that here. And as I said, it's also an important cultural phenomenon. We have historical evidence that midwifery has been important to all kinds of human cultures for a long time. And in a way, what we see is really a sort of gene-culture coevolution. So the midwifery, the active assistance during childbirth is really part of also of our biology, because without that it wouldn't work properly. It would even work less. And this active assistance is also unique to humans. And in a way, it's itself an interesting biological phenomenon, which remembers to the Bogart effect, because at the one hand, midwifery enables or eases childbirth. At the other hand, it hinders additional evolutionary adaptation, because without midwifery, the mortality rates would be greater and hence the selection stronger. So the anatomy would have adapted better. But sort of that is a two-sided thing here. But it's obviously super important and really part of our biology, so the midwifery and obstetric tradition. And as you also know, modern obstetrics, especially C-section, is highly variable globally. So we have C-section rates from a few percentage up to more than 50% globally. But even in Europe, for example, which is culturally more homogeneous, we have C-section rates from about 40–50% in Scandinavia to 35 or more percent in some southern and southeastern countries. So that's interesting in itself. So, and I can't go into these topics here, but there's a lot of research going on that, and it's very interesting. But what we clearly have is a situation where we have biological, anatomical, genetic influences. We have social factors, cultural factors, individual psychological factors. We know that, you know, on Friday evenings there are much more C-sections than on Monday mornings for obvious reasons. So it's really a complex phenomenon with many layers. And the challenge is how can we study it, and where can we cut this complex system into pieces to be studied separately? Yeah? And because of this complexity, several authors have argued actually that there is no obstetrical dilemma. In the sense that childbirth isn't that difficult and there is no evolutionary history of antagonistic selection. That the alleged difficulty of childbirth mainly resides from inappropriate obstetric practices and lifestyles, as well as patriarchal cultural and medical systems. They argue that the evolutionary narratives of an obstetrical dilemma are problematic because they foster pathologization and over-medicalization of childbirth. I understand that motivation, but I disagree. And just a few months ago, we have published with a number of scholars in this regard a paper called "There is an Obstetrical Dilemma," where we argue that even though we share the political intention that childbirth isn't pathological and C-sections shouldn't be practiced too much, it shouldn't be over-medicalized, that's all clear, but there's very strong evidence of this antagonistic evolutionary history. And we argue that evolutionary and morphometric studies need not hinder appropriate and evidence-based midwifery and obstetric care, nor do they promote negative use of women's bodies and capability, because some have argued that telling about an evolutionary compromise implies that women are compromised, which we disagree with. I mean, that is really different. And we believe that understanding the evolutionary entanglement of biological and social cultural factors can help us to understand not only individual variation in risk factors of obstructed labor and thus contribute to a more individualized and less invasive obstetric care, but it can also be important for the long-term planning of public health and research policies. For example, in in our model, what increases this evolutionary part of the increase in cesarean section is driven by the selection for large babies and narrow birth canals. And of course, also this has been largely reduced in modern societies because of advances in neonatology and pelvic floor surgery. But you know that this relationship that if you reduce maternal mortality through C-sections, that you should also invest into the research and practice of neonatology, so the care of newborn babies and pelvic floor surgery, because this reduces also the selection, so it reduces the evolutionary increase of obstructed labor. I think that's important to understand for public health policies and also for directing research directives. And I think that this doesn't only apply to the obstetrical dilemma. I've also argued that many other diseases, including, for example, autoimmune disorders, might be subject to similar dynamics. Okay, with that, I thank you for your attention. Thank you for all that, for the medal, and for all these nice words and everything. So thank you very much.